Corinne Bertolotto is our next speaker. Moving now from hematopoiesis to solid tumors and post-translational modifications.
Good afternoon, everyone. So first, I want to thank Hugues for the invitation. It's a pleasure to be here. And today, I'm going to talk about the transcription factor MITF in melanoma and its deregulation via Sumoylation. So melanoma is a deadly cancer. Melanoma is a deadly cancer due to its high metastatic propensity. It represents less than 5% of skin cancer, but is responsible for 80% of deaths by cutaneous cancer. Its incidence has been rising during the last 30 years. It's about 14,000 new cases diagnosed each year in France and about 320,000 new cases worldwide. In recent years, major breakthroughs have been realized in the treatment of metastatic melanoma with newer types of targeted therapies, which is with vemurafenib or PLX4032, and immunotherapies such as the immune checkpoint inhibitor, anti-PD-1, and anti-CTLA-4. So here you can appreciate the efficiency of the targeted therapy with vemurafenib that targets the oncogenic form of BRAF. So you can see that 15 weeks after the beginning of its treatment, the MET here almost completely shrunk. However, 7 weeks later, it relapsed. And usually relapses are more aggressive and invasive. So melanoma is still a fatal disease, and we need to better understand the molecular mechanisms to design more effective treatments. And so far, early detection remains the best way to cure melanoma. So what do we know about the molecular mechanisms involved in melanoma? So melanoma can originate from a melanocyte, a cell of the skin, which physiological functions is to synthesize the melanin, the pigment that are responsible of our skin color. This is in 75% of the cases, but in 25% of the cases, melanoma can originate from a pre-existing nevus or mole. The nevus is a benign lesion that is thought to be formed of melanocytes that enter the process of senescence upon oncogenic activation, such as oncogenic BRAF and NRAS, which are the main alterations found in melanoma. So here on the right, you have staining of normal skin and nevus sections. You can see that the nevi are positive in brown for the cell cycle inhibitor, P16 in 4A. That means that these lesions are growth-arrested. And below, the blue staining represents the senescence-associated beta-galactosidase staining, which means that these lesions are growth-arrested in senescence. So more than 80% of common nevi or congenital nevi harbor oncogenic mutation in BRAF or NRAS, respectively. This suggests that these oncogenic mutations are not sufficient by themselves to induce the malignant transformation of melanocytes. So the nevus has been posited as one of the best examples of senescence in vivo, and it can remain static for decades, never progress to malignancy, unless other alteration suppressing senescence occurs. So this can be the loss of CDKN2A, which encodes P16 in 4A, we just saw, or the loss of P10, or amplification of genes that control the cell cycle, such as CDK4 or cyclin D1. Senescence bypass allows melanocytes to restart proliferation. You know that during cell divisions, errors can occur, leading to genomic instability, uncontrolled cell proliferation, and with acquisition of a pseudo-epithelial mesenchymal transition program, melanoma cells can start to disseminate, form distant metastases that usually are drug-resistant. So senescence bypass and invasiveness and drug resistance are the two steps to target if we want to prevent melanoma progression and invasiveness and drug resistance. In melanoma, drug resistance can be driven by genetic events, but the phenotypic plasticity that allows change in epigenetics and rewiring in metabolisms is now accepted as the main source of resistance. And basically, melanoma are composed of two main subpopulations of cells, the proliferative and invasive cells. And so during tumor progression or under stress conditions such as hypoxia, nutrient starvation, or cytotoxic drug effects, cells are able to switch back and forth between these two phenotypes, thereby creating intratumor heterogeneity that may impact on the therapeutic effect. So identification of the mechanisms involved in this phenotypic switch is mandatory to develop effective and long-lasting therapies. And we now know that the transcription factor MITF is a key actor in this phenotypic transition. So MITF stands for microphthalmia-associated transcription factor. This is a master gene of melanocyte homeostasis, and you easily understand why, because here you can see that MITF loss of function mutation in both mouse and humans triggers loss of the melanocytic lineage. And for example, this mouse is not white because it has non-functional melanocyte, but because it has no melanocyte at all. So in melanoma, the role of MITF is complex, and this is a sort of sum up. Proposed a RESTA model proposed by the colleagues years ago, Colin Godding in UK. So MITF is controlled at the transcriptional level through transcription factors such as Krebs, Soxten, beta-catenin, also via gene amplification. MITF can also be controlled at the post-translational level through phosphorylation, Sumoylation, and caspase cleavage. And this regulation impact on MITF binding to cofactors such as the histone acetylase CBP or the chromatin remodeler BRG1. And this regulation creates a sort of barcode-like situation that channels MITF towards a specific subset of targets and determines its biological activity. So basically, cells with low level of MITF are in a senescent and stem cell-like state that is associated with an invasive and inflammatory signature characterized by increased expression of tyrosine kinase receptor, AXL, PDGFR beta, increased expression in the inflammatory cytokine IL-6 or the epithelial mesenchymal transition factor ZEB1. And this invasive signature has been associated with therapy resistance. Cells with high level of MITF are in a proliferating and differentiated state associated with a proliferative signature characterized by increased expression of genes that control the cell cycle, such as CDK2 or pigmentation genes. So regarding caspase cleavage, to make a long story short, years ago we identified MITF as a new substrate of caspases during induction of apoptosis in melanoma cells. And what we show in this study is that the non-cleavable form of MITF render melanoma cell resistant to apoptotic stimuli. And this prompts us to hypothesize that individuals who develop melanoma harbor a mutation in the caspase cleavage site. And to assess this hypothesis, we collaborate with Brigitte Bressac de Pailleret, who is in the audience, and we haven't found so far a mutation at the caspase cleavage site, but what we found is a mutation, a germline mutation that changes the glutamate free into lysine. So you can see here that the frequency of the E-free-18 lysine variant is higher in patients with single or multiple melanoma compared to control. This variant has been also identified by other groups with similar allele frequency in patients with a single, multiple melanoma, but also in cases of familial melanoma. And in this study, the variant has been associated with nodular melanoma, which is a form of aggressive melanoma. The variant confers to carrier a 2 to 5-fold increased risk of developing melanoma. And what is interesting also to note is that this variant has been associated with fair skin and increased nevus count, which are melanoma risk factors. So the first question we have addressed is the impact of this mutation on MITF. This is here a schematic representation of MITF, which is a basic helix loop helix leucine zipper transcription factor. So here with the main post-translational modification, phosphorylation, ubiquitination, and 2 Sumoylation sites were identified. And you can appreciate that the mutation we identify in patients is located near the second Sumoylation site. This is here sequence alignment, and you can see that the glutamate free is conserved among species ranging from human to fish. So the change of glutamate free into lysine change the consensus simulation site IKQE into IKQK. That means that this mutation can impact on the simulation of MITF. And this is what we show here. This is an experiment performed in Nectin-3 cells in which we co-express SUMO1 and MITF. This is another exposed blot. So you can appreciate that MITF is detected as a doublet, the upper band, being a form phosphorylated on serine 73 by the MAP kinase pathway. So we observed 2 simulated forms when we perform the mutation of the first simulation site, we decrease the simulation of MITF. When we introduce a mutation, we identify the patient, we even more decrease the simulation of MITF. And when we perform the double mutation, we lose the simulation of MITF. So this means that the E318 lysine mutation impairs the simulation of MITF. Recently, we confirmed these results in a more physiological setting without forced expression using proximity ligation assay with anti-SUMO1 and anti-MITF in melanocyte isolated from patient with wild-type MITF or in a melanocyte isolated from patient with a MITF mutation. And this is here immunofluorescence showing, this is a magnification here, showing the dots here that reflects the interaction between SUMO and MITF that were detected in the melanocyte with wild-type MITF, but there is a dramatic reduction of this detection in a melanocyte with a MITF mutation. So having shown that the E318 lysine mutation impairs the simulation of MITF, we next address the impact of this mutation on the biological activity of MITF. So we observe no change in the cellular localization, no change in the stability of a MITF mutant, but what we observe is a change in its transcriptional activity, and this is what is shown here on this slide. So this is an experiment performed with a synthetic reporter vector comprising 3 e-boxes, which are the site recognized by MITF, in tandem upstream the luciferase genes. So here you can see that the wild-type form and the mutant and the mutation in the first simulation site display similar transcriptional activities. But when we introduce the mutation we identify in patient, we found a statistically significant increase in the transcriptional activity of MITF. We also determined the global repertoire of the wild-type and the mutant form of MITF using chromatin immunoprecipitation coupled to high-throughput sequencing. So what this experiment tells us is that about 9,000 sites are bound by the wild-type form of MITF and about 22,000 by the mutant form of MITF, the difference being a large number of weakly occupied sites. So comparative analysis of the groups using read density matrix clustering, which is shown here, reveals that in some clusters, such as cluster E, for example, sites are equally occupied by the wild type and the mutant form of MITF, but in other clusters, such as in D, for example, sites are preferentially occupied by the mutant form of MITF. On the right, you have example. This is UCSC genome browser view of the ChIP-seq profile of the mutant and wild type MITF. So you can see that there is a higher occupancy of the IF1 alpha, AP1, or IBCB5 loci by the mutant form of MITF compared to the wild type. So these experiments indicate that the MITF mutation modifies the DNA binding activity of MITF. So further to that, we use the MIM program to analyze the motif bound by the wild type and the mutant form of MITF. So for that, we use the top 500 sites specifically bound by wild-type MITF or by the mutant form of MITF. And what this analysis tells us is that the wild-type MITF binds to a 6 base pair E-box motif, whereas the mutant form of MITF has a preference for an extended 8 base pair palindrome. This means that perturbation of MITF simulation, such as the mutation E318, the mutation of the glutamate freighting into lysine, impact on the genomic distribution of MITF. So having shown that the E318 lysine mutation induces MITF Sumoylation, change MITF genomic distribution and its transcriptional activity, the next question we address is a link between the MITF mutation and the pathogenesis mechanisms. And for that, we constructed a new knock-in mouse model expressing the MITF mutation from its own promoter. So these mice did not show growth abnormalities. They didn't show any sign of nevi or melanoma within 24 months after birth. The only phenotype we observe is a slight lightening of the pigmentation of the coat and the skin of these mice. And here you have a Fontana-Masson silver staining showing in dark here the pigment in the tail skin of the wild-type mice and the dramatic reduction of this pigment in the tail skin of the mutant mice. This is here on the right, the quantification of the reduction in the pigmentation. So to elucidate the importance of MITF mutant in melanoma occurrence in vivo, we next sought to introduce the MITF mutation in 2 mouse models prone to develop melanoma. So the first model we used is the model that allows expression of oncogenic BRAF, specifically in melanocytes after tamoxifen application. So this mouse model is known to develop, to form nevi, and this is exactly what we observed. This is the presence of nevi after tamoxifen application. And what is interesting is that when we introduce a MITF mutation, we observe a statistically significant increase in the number of nevi, indicating that the MITF mutation is nevogenic. And this is in agreement what was reported in humans with the MITF mutation. Regarding melanoma development, this is a Kaplan-Meier analysis. Also, we observe a tendency to a shortened survival with the MITF mutation. The difference between the two groups was not statistically significant. So we introduced a MITF mutation in a second mouse model. This mouse model allows expression of oncogenic BRAF and the loss of PTEN, specifically in melanocyte upon tamoxifen application. So here you have a representative kinetics experiments. And what we observe is that in presence of the MITF mutation, the lesion develop earlier and progress faster to melanoma. And this is here the Kaplan-Meier analysis, and we found indeed a shortened survival that is significantly different between the two groups. A shortened survival of the mice in presence of the MITF mutation. So to understand the molecular mechanisms, we perform a transcriptomic profiling of 4 different tumors from these two groups. And what this analysis shows is that the MITF mutation is associated with a decrease in expression of genes that control the senescence process and that are important for this process in melanocytes, such as CDKN2A and 2B, indicating that the MITF mutation impairs the process of senescence. And this is what you can see here on these experiments that we perform with melanocytes isolated from patients with wild-type MITF or with a mutant form of MITF, that we transduce either with an empty vector or an oncogenic form of BRAF. You have here the control of the experiments. And so as expected, wild-type melanocytes enter the process of senescence upon oncogenic activation, but there is a dramatic reduction of this ability to enter senescence in the melanocyte with a MITF mutation. So the MITF mutation impairs oncogen-induced senescence in melanocytes. So at this step, this is a model I would like to propose. In contrast to MITF wild type, which is a pro-differentiation factors, and which also is known to stimulate the proliferation of melanocytic cells, the MITF mutant form, inhibits the differentiation programs and is able to induce a bypass of the process of senescence. And what we show also is that these melanocytes have a shorter doubling time compared to a melanocyte with wild-type MITF, and so better stimulates their proliferation, and also favors the transition towards melanoma. So of course, because the MITF loss-of-function mutation inhibits the development of the melanocytic lineage, and because MITF is a key factor in the control of melanoma cell proliferation, years ago we also asked the impact of MITF suppression because we thought that MITF was a good candidate to pursue at the therapeutic targets. And so for that we used a siRNA approach. So here you can appreciate the efficiency of the siRNA against MITF over time. What you can see also is that in contrast to control cells, which are transfected with a control siRNA, MITF suppressed cells stops proliferating. MITF suppressed cells also adopt an enlarged and flattened morphology and are positive for the senescence-associated beta-galactosidase activity. So usually senescence is associated with induction of DNA damages, and here this is a brief representation of the pathway. So induction of DNA damages induce recruitment of DNA damage sensor, such as gamma H2AX and 53BP1, that creates a platform to recruit a DNA damage effector, such as ATM and CHEK2 that are activated by phosphorylation, leading to stabilization of P53. And P53 coordinates the cell cycle arrest and DNA repair, if it is possible. Otherwise, cells undergo senescence or apoptosis. So here, by immunofluorescence, we observe that a MITF suppressors displays a recruitment at site of DNA damages of gamma H2AX, 53BP1, and activated ATM and CHEK2. And here, by immunoblot, you can see that in MITF suppressors, we have a phosphorylation of P53 on serine 15 that is known to induce its stabilization and an increase in the level of P53. So what we observe is that MITF silencing induces classical hallmark of cellular senescence, but no apoptosis. So I show you that MITF suppression induces the senescence program, and actually the cells are arrested in a G0G1 group phase. But when we perform subcutaneous tumor with these cells, we found that the MITF suppressors gave rise to tumor bigger than control cells. Further to that, we analyzed these cells. You can appreciate the efficiency of the MITF siRNA, and we also observe the decrease in the expression of two MITF target genes, tyrosinase and SLUG. But interestingly, we observe an increase in the stemness marker, snail, and fibronectin-1, in the mesenchymal marker, snail and fibronectin-1, and in the stemness marker, OCT4 and NANOG, indicating that the loss of MITF also stimulates the tumorigenic potential of melanoma cells. So the next question we had addressed is whether there is a naturally occurring population expressing very low level of MITF. And so by FACS, we analyzed the expression of MITF in different cell line and short-term culture. And this is here an example. And you can see that indeed we found a small population, about 2% of cells, that express very low level of MITF. So we next wanted to address the tumorigenic potential of the MITF high and low population. But MITF is a transcription factor exclusively localized at the nucleus. And there are no surface markers allowing to distinguish the high and low MITF population. So we used another approach based on CFSC, which is a vital dye whose fluorescence intensity decreased by half at each cell division. So here, this is the initial loading of the cells with the CFSC, and the cells were let to divide in culture. So here, this is the CFSC high population with cells that didn't divide or divided only one time, and here the CFSC low population with cells that divided several times, 1, 2, 3, 4, 5, etc. So we analyzed MITF in the CFSC high and low population, and what we observed is that the CFSC high population contained a high proportion of MITF low cells, whereas the CFSC low population contained a low proportion of the cells expressing low level of MITF. And when we perform xenograph experiments with the CFSC high and low population, this is 4 different xenographs, we observe that only the CFSC high population is able to form subcutaneous tumor. So this means that the MITF low population is required for melanoma tumor formation, and these MITF low cells display characteristics of melanoma initiating cells. So to sum up this part, I show you that MITF inhibition triggers senescence and stem cell-like phenotype that enhance the tumorigenic potential of melanoma cells. And I have no time to show you, but we also observed that the reduction in MITF enhances the invasiveness and metastasis ability of these cells. So of course, the quick question is, what are the signals that decrease MITF level and trigger this invasive phenotype? And today I would like to share you a very recent data. So we were recently interested in determining the impact of vemurafenib on the metabolisms, on the global metabolisms of melanoma cells. And for that, we perform a global metabolomic approach using UPLC mass spect in 2 distinct melanoma cell lines, expressing the oncogenic form of BRAF in control condition and after vemurafenib exposure. And so we analyzed 500 metabolites, 60 were up, 60 were down. This is here the heat map. And our attention was drawn here by the decrease in the level of the nicotinamide adenine dinucleotide and the global reduction in the nicotinamide pathway, because NAD play a key role in energy production and in numerous cellular function by controlling enzyme activity. And to make a long story short for this part of the publication, what we show is that BRAF controls the nicotinamide phosphorylated transferase NAMT at the transcriptional level, and NAMT control the NAD synthesis in melanoma cells. So the next question we had addressed is the impact of NAD and NAMPT on the response to PLX in melanoma cells. And for that, we force the expression of NAMPT in two cell lines. So you can appreciate the increased expression of NAMPT that translated, we confirm, in an increased NAD level, of course. And what we observed is that forced expression of NAMPT reduces the reduction of the cell number mediated by PLX, by vemurafenib. And here on the right part, this is we had exogenous NAD to the culture medium, and we also observed that exogenous NAD prevents the reduction of the cell number mediated by PLX. So this means that the NAMPT and NAD metabolism induce drug resistance in melanoma cells. So we next wanted to elucidate how NAMPT and the NAD metabolisms can confer this BRAF inhibitor resistance. And so for that, we performed transcriptomic profiling in 3 different melanoma cell lines in control condition or after NAMPT expression, force expression. So we found about 1,000 genes differentially expressed. Here you have the heat map. Engineering pathway analysis reveals striking increase in cellular function associated with cell movement and migration of the cells. And what you can see here is that cells with forced expression of NAMPT display an increased expression of genes associated with invasive signatures such as ZEB1, AXL, or PDGFR-beta, and a decreased expression in genes associated with a proliferative signature such as MITF and CDK2. Further to that, we used the IPA upstream regulator tools to identify potential upstream regulator of this signature. So we observed that TGF-beta, NF-kappa B, or SMARCA4 were increased, and the MITF was decreased. So this means that NAMPT favors the transition towards an invasive phenotype. Interestingly, SMARCA4 is BRG1. It is, I told you that it is a binding partner of MITF, and it is also a key chromatin remodeler. So this suggested that NAMPT can impact on the epigenetic landscape in melanoma cells. So to assess this hypothesis, we perform ChIP-seq experiments of the main activating and repressing histone marks. So we found 40,000 peaks within 12 clusters. Cluster 1 to 6 out of the 12 show alteration in these histone marks. So for example, you can see that we have an increase in activating histone marks in cluster 3 and 2, a decrease in activating histone marks in cluster 4, and an increase in repressing histone marks in cluster 6. Here, this is example of the ChIP-seq experiments at the MITF and AXL locus. And so you can see that after forced expression of NAMPT, we have a decrease in activating histone marks at the MITF locus. And here, the stimulation of the activating histone marks at the AXL locus. So taken together, the transcriptomic experiments and the ChIP-seq experiments, this suggested that NAMPT control the motylability of melanoma cells. And this is what we show here. We use Boyden chamber experiments, and cells with forced expression of NAMPT display a higher motylability compared to control cells. Further to that, we analyzed these cells. So we found that the decrease in MITF, the increase in NAMPT expression is associated with a decrease in MITF and an increase in the mesenchymal marker fibronectin 1. And here, the decrease in NAMPT is associated with a decrease in ZEB1, which is a key factor involved in the motylability of melanoma cells. And finally, so what we show also is that targeting NAMPT is a valid therapeutic option in melanoma, and this is here an example of an experiment performed in BRAF inhibitor-resistant melanoma cells. So they are indeed resistant to PLX, which is vemurafenib, but these cells remain sensitive to the effect of FK866, which is a NAMPT inhibitor, and when we combine PLX with a low-dose FK, we have a synergistic effect. So NAMPT and NAD pathway alters the epigenetic landscape, and inhibition of NAMPT can be a therapeutic option in melanoma. So to finish, this is a model I would like to propose. So I told you that MITF is a pro-differentiation factor and stimulates the proliferative activity of melanocytic cells. I show you also that it prevents the transition towards the melanoma-initiating cell phenotype. The mutant form of MITF is block the differentiation program, favor senescence bypass and proliferative activity of these melanocytic cells, and contribute to favor melanoma progression, whether it has a role in the transition towards the melanoma initiating cell phenotype is under current investigation in the lab. But I show you that we have a higher occupancy of the ABCB5 locus by this mutant form of MITF compared to the wild type. So what I show you also is that NAMP and NAD metabolisms can inhibit MITF and favor this switch towards the melanoma initiating cells and melanoma development. And I have no time to show you, but we also show that the MITF level can be controlled by hypoxia and by the senescent secretum produced by the senescent melanoma cells. So I would like to finish by thanking all the people in the team who did the work. So of course, Robert, and the former PhD and postdoc, Michael, Caroline, and Jean-François. And this is our collaborator that I would like to thank, and especially Brigitte, who is in the audience. Thank you very much.